Hello everyone, and thank you for being interested in our work, aiming to explore brain mechanisms supporting motor skill learning and the consolidation of new motor memories. I have no financial interest or any relationships to disclose. Learning new motor skills meets us at every stage of our lives. But how the learning of a new motor skill, such a fundamental ability in our lives, is eventually achieved? What changes occur in a brain that enables us to perform that new piece we have just learned and practiced on the piano even better the next day? And which brain mechanisms in turn support and promote these changes in the human brain? The learning process does not end when the practice session is finished. Instead, a variety of neural processes which take place following practice or offline and are generally termed consolidation processes are initiated and operate on different brain scales and time scales across both wakefulness and sleep following the learning experience, with some initiated even immediately following the online practice. These processes in turn act to promote two main basic aspects that support the new learning: the offline enhancement of skill performance after the practice has already ended, and the stabilization of the newly encoded neural representation of the motor skill or the motor memory. The primary motor cortex, or M1, plays a vital role in different stages across the motor learning process. Learning-induced structural and functional changes have been documented in M1 following motor learning in both humans and animals, ranging from micro to macro scale structural and functional changes, including the formation of a neural representation of the motor skill memory, or a motor memory engram. Also, M1 is part of a cortico-striatal network that subserve motor skill learning. Previous studies in animals have shown that glutamate and GABA, the main excitatory and inhibitory neurotransmitters in the brain, respectively, may play important role in the neurobehavioral adaptation to learning, and recent studies in humans have provided evidence for a role of excitation and inhibition in M1 during the online phase of motor skill practice. However, it is still unclear what role, if any, changes in glutamate and GABA following learning may have in promote motor memory consolidation and motor learning-induced plasticity in the human brain. Therefore, in the current study, we aimed to investigate whether short-term changes in the concentration of glutamate and GABA within the first 30 minutes following motor skill learning may be associated with longer-term learning-induced neurobehavioral plasticity as measured overnight in terms of improvements in behavior or in skill performance, changes in the functional communication of M1 with the striatum, a key region in motor skill consolidation, and changes in M1 gray matter volume. To answer these questions, we conducted a repeated measure study across 2 consecutive days, in which participants engaged in a well-known motor sequence learning task inside the MRI scanner and were learning to perform a tap-in sequence as fast and accurate as they could with their non-dominant hand. On the second day, participants were then tested for their performance of the skill. Neuroimaging measurements were conducted on a 7T scanner. Neurochemical measurements were acquired with a semi-laser sequence recently optimized in our lab, and a 2 cm cube spectroscopic voxel was placed in the contralateral M1 of the learning hand. MRS analyses were conducted using the VDI libraries and LC model, and we acquired a baseline measurement at the beginning of each day and 4 additional measurements during the first 30 minutes following the learning task on the first day. Functional data were acquired using the Human Connectome Project protocol with a TR of 1 second and isotropic voxels of 1.6 millimeters. We also used spin echo maps with opposite directions to correct for the EPI distortion, and pre-processing the noising of the data and higher level analysis were conducted with FSL and the Con toolbox. Structural data were acquired using an MP2 reg sequence with isotropic voxels of 1 millimeter and were analyzed using the voxel-based morphometry or VBM pipeline that is implemented in FSL. ROI ROIs of M1 and the putamen for the functional connectivity and structural analysis were defined as the voxels within those regions which demonstrated significant task activation during both the first and second day. This definition is based on the definition of memory engram cells, which are activated by the learning experience, the initial learning experience, and are further reactivated by subsequent retrieval of the learned memory. Also importantly, all the M1 ROI voxels were located within the boundaries of the MRS voxel. So, what have we found? First, although we did not observe a group-level change in either glutamate or GABA following learning, despite a trend toward increased glutamate after 30 minutes, we did observe a significant correlation between greater increases in glutamate following the learning task and overnight improvements in skill performance or adaptive behavior. Next, we found that greater increases in glutamate immediately following learning were linked to overnight increases in the functional connectivity of M1 with the right putamen, which in itself was associated with increased task performance, attesting for the importance of this measure in motor skill consolidation. Lastly, we found that greater reduction in GABA after 30 minutes following the learning task, but not immediately following the task, was associated with overnight increases in M1 gray matter volume, although we did not observe a relationship between those structural changes and changes in task performance overnight. It is also important to note that in autocorrelation analysis, we controlled for the changes in the other metabolite. We also did not find a significant relationship between any neurochemical changes and changes in the functional connectivity between M1 and the control region, the posterior cingulate cortex, for either glutamate or GABA. Also, neurochemical modifications were not related to the extent of neurobehavioral activation, not in terms of the number of key presses during the MSL task, or in terms of the beta value of the MSL task blocks. So, what can we conclude from these findings? First, since in each correlation analysis we controlled for the change in the other metabolite, our results suggest an independent role of glutamate and GABA in different neural and behavioral aspects of motor memory consolidation and motor learning-induced plasticity. In addition, our findings suggest that glutamate and GABA-related neuroplastic changes may operate on different timescales, maybe starting immediately following learning in the case of glutamate and expressed later in the case of GABA. Thank you very much for listening and have an enriching conference.